If I could have the speakers for the next panel, the Emerging Therapies panel, to please come up and take a seat, that would be great. While folks are coming up for the next panel, I want to make one announcement. Unfortunately, in my opening remarks when I was talking about resources for the FA community, I missed a really important one. There is a Facebook group for individuals with FA only. There are not, other family members, friends, or anyone else on the FAR only group. There are over 700 individuals living with FA that are a part of that group and contribute to supporting one another. And so that is an excellent resource if you are on Facebook and want to connect with more community members. And Norm Simpson, thank you for that reminder. All right, so we are going to start our next panel. And our next panel is going to transition us to talking about therapies, to solving our challenge of no approved therapies for FA. And we'll actually have two panels. The first one is for focusing on treatments that are not in the clinic yet. And so we're calling that the Emerging Therapies panel. And then the second panel, after we have a short break, will be on treatments that are currently in clinical trials. So I'm going to give a little bit of introduction to both panels right now to get us started, and then I'm going to turn it over to our speakers. So the first polling question before we get started with this panel asks you what you think therapeutic targets for FA include, and I have a list of potential ones here. I think everybody's catching on. The good news is that there are a lot of therapeutic targets for FA, and we're going to talk about many of them today as each of the speakers presents. Different therapeutic strategies are coming forward, and the exciting thing is that they work in different ways. They might target different aspects of the underlying disease. If I could get the slides back. So just to make sure we're all starting from the same foundational knowledge before I turn it over to our speakers, just a few of the sort of the basics, the underpinnings of FA that get us to those therapeutic targets. So I think one of the questions we had from online was, what's a genotype and a phenotype? So the genotype is your underlying genetic information or your genetic profile. And in Friedreich's ataxia, the genotype that causes the disease is the same in 96% of individuals. So there's what we call genetic homogeneity or genetic sameness. And 96% of individuals with FA have this trinucleotide repeat expansion in the first part, or this first intron of the FA gene. The FA gene is also known as FXN for short, and the GAA is just the genetic code. It's the DNA, and it repeats itself, GAA, GAA, GAA. We all have GAA repeats in our FXN gene at this specific location. It's just how many repeats that distinguishes an FA allele from a non-FA gene. When the number of GAA repeats is less than 30, the gene is on. That's what we're saying. The gene's on, and it can make a protein called frataxin. When there are expanded repeats, the gene gets shut down, and you don't make enough frataxin protein. So let me back up one more step. Just a few basic reminders from high school or college biology. So this is a cartoon of a gene, and genes are made up of exons and introns. The exons are the coding sequences for the genes. The introns are also made up of DNA, but they don't actually code for the protein. All genes, and another sort of easy way to think about this is think of a gene as a set of instructions. and the gene has to get read in order for the gene to make the desired protein. But there's a whole process by which this occurs. The reading of the gene is called transcription. There's an intermediary where these introns get spliced out, and you have a product that's called mRNA. And that mRNA is an important set of instructions coming from the original gene that actually get translated into protein. And that happens for every single gene in the body. It's not unique to FA. Every gene gets transcribed to mRNA and then translated into protein. So that's how we get from the frataxin gene to the frataxin protein. And what's happening here when there are expanded repeats is that transcription process gets shut down. And so at the beginning, it's just not able to get read. There are a few important things to keep in mind about this transcriptional deficiency. One is that it's not 100%. It's not completely shut off. So people with FA make some frataxin protein, just not enough. And in this graph, what I'm showing you, the darkest gray or black bar here are control individuals. And let's just say control individuals make 100% of the cells required for frataxin. Over here in the light gray are individuals with FA. And there's a range of frataxin deficiency, even within people with FA, tends to be about 4 to 20% of normal. And then individuals who are carriers, so parents and some siblings who have one copy of the gene with expanded repeats and one copy with normal repeats, make about 50% of the normal amount of frataxin. And we know that carriers don't have symptoms of FA. And so when we think about treatments, there are a few things that are encouraging for us. One is that we don't have to get people from this 5 to 20% to 100. We only have to get people probably up to 50% for their cells to be able to function properly in the face of frataxin deficiency. Also, again, it's not that the gene is completely shut off and that there's no frataxin. There is some there. And so there are multiple ways that we can start to think about how to elevate frataxin, and that's what you're going to hear about in some of the next presentations. So what does frataxin protein do? Frataxin is a highly conserved protein. Turns out yeast make frataxin. The most simplest of organisms make frataxin to the most complex. It's highly conserved. That's what highly conserved means. Evolutionarily, it's highly conserved protein. And it functions in an important part of the cell called the mitochondria. And the mitochondria are where the cell gets its energy. And so this is a cartoon of a cell. This nucleus part here in the middle is where the DNA is. That's where the frataxin gene is. The gene gets transcribed, the protein gets made, and these little organelles within the cell, these ones right here are the mitochondria, they take up that frataxin protein. The frataxin protein in the mitochondria has an incredibly important role. We now understand it is part of a complex in that mitochondria that makes something called iron sulfur clusters. And we're not going to become, biochemists today, but the point is that these iron sulfur clusters are essential for lots of different processes in the mitochondria and outside the mitochondria. And the reason why we have that long list of potential targets for treatment is because iron sulfur clusters are essential for lots of different things. And so if you don't have frataxin or enough frataxin, you also don't have enough iron sulfur clusters. And if you don't have enough of those iron sulfur clusters, there are downstream consequences. And we understand a lot about that now and can target those 4 potential therapies. And that's why we've got such a great opportunity for multiple different therapeutic approaches. So hopefully what you're getting a picture of now is sort of a cascade. And we can start at the gene itself, the frataxin gene, and the repeats. And we can start to think about different targets around the gene that might be gene replacement or gene editing. Can we cut the repeats out? Can we target transcription or translation and try and turn that back on in some way? Can we target the mitochondria themselves and look at ways to improve mitochondrial function, even if there's frataxin deficiency? And then we can think about what we understand at the organ tissue level, like what you just heard in the previous panel when we think about diabetes and heart disease, and can we come up with symptomatic therapies from other more common conditions and apply those? So we have multiple therapeutic approaches, and this is one of the things that makes me excited and optimistic for the future and the ability to be able to treat FA is the fact that we have multiple therapeutic targets. I just want to make sure everybody's familiar with the treatment pipeline. This is on our website. We try and keep this updated almost on a sometimes daily basis, but at least monthly basis. And you can get a lot more information about what's going on in terms of treatment development from the pipeline. But just to orient you, so going down the left side, we start with treatments that focus on the mitochondria, then the downstream, or the metabolic pathways that are affected in FA, frataxin protein itself, therapeutic strategies that look at trying to get more mRNA or more gene expression that would then get us to more protein, and then gene replacement and gene editing. So these are the sort of approaches to therapies, and then across the top is how far this therapy has gotten in development. And so starting over here on the left are things that are very early discovery stage, to preclinical, which means it's still testing in the lab, it hasn't advanced to human testing. An IND submission is a regulatory milestone that has to be met to move from the lab to the clinic. And then here we have the stages of clinical development, phase 1, phase 2, phase 3. These are the different clinical testing phases that we go through, clinical trials, and then ultimately to filing of a new drug application and hopefully having therapies available through, doctors prescribing them. We are going to have a separate talk later about the regulatory process, so you're going to learn more about that later in the day. So just to kind of sum up, where we are, you're going to hear about a lot of different treatment approaches. So there's diversity in how we're trying to think about treatments for FA. And we have candidates that are very early in discovery, as well as very late, almost ready to be filed for approval. So we have some near-term therapies and some therapies that are still early stage that are coming along. So lots of opportunities for drug development, but also for you to be involved. Because as things move from the lab to the clinic, we need people to participate in clinical trials. And so with that, my last polling question to get you starting to think about clinical trials is, which of the following risk factors would influence, or not risk factors, sorry, which of the following factors would influence your decision to take a new medication or to participate in a clinical trial? And there are multiple different things that are listed here, but I ask you to take a minute to think about this and select the top 3 factors that are most important to you. Would it be risk of serious side effects, risk of common side effects? Oh, and now I can't read the screen. How the treatment's administered, the burden of administration, what might be involved in the trial in terms of number of visits or the frequency that you have to go to the hospital, if you had to change something that you currently do, the potential benefit that you might get from participating in the study or the medication, the impact to participate in future trials, cost, travel, or something else we haven't thought of. And we'll share the results of all the polling. Afterwards, I realized when we set the polling questions up, the percentages are coming up a little funny because we didn't factor for letting you select multiple responses, so we'll fix those before we share them. But I think the bars do reflect the frequency with which people are generally responding. And so, I think it makes some of the results we're seeing make a lot of sense in that risk for serious adverse event would definitely influence decision about taking a new medication, whether or not you have to change your current treatment, the potential for benefit, and how this might impact your ability to participate in future trials seem to be the top 4. So thank you for taking the time to answer this question. I think this is not only helpful for us, but certainly for our partners who are here today to hear from you about kind of how you think about things when it comes to clinical research and taking new medications. So with that, our first speaker couldn't be here in person today. We have a recorded presentation from Avanti Bio. Dr. Chris Wright is their Chief Medical Officer. And so he sent his presentation for us to view. And our AV team should be able to roll that for us now.
Thank you so much for the opportunity to speak with you a bit about Avanti Bio and our Friedreich's Ataxia program. My name is Chris Wright, and I recently joined Avanti as CMO. I'm a neurologist and neuroscientist by training, and I also saw patients for about 20 years, mostly while I was also working in the biotech industry. At Avanti, we're focused on developing new genetic therapies for patients in need. We have a great team that's focused on moving our programs forward, and the company is supported by top-notch investors who provided us the resources needed to move our research forward into the clinic. And as I'll discuss today, our lead program is an FA. The key funding came to the company in 2020, while the company was initially incorporated a year earlier in 2019. We're headquartered in Cambridge, Massachusetts, and have a major presence also in the Gainesville, Florida area, where we have a key strategic partnership with University of Florida, which is also where our founders are from and where some of our early finance support came from. These are our 2 founding scientists, Dr. Barry Byrne and Manuela Corti. Dr. Byrne is the head of the UF Powell Gene Therapy Center and has been a scientific leader and clinical pioneer in the gene therapy area for many years. Dr. Corti has extensive experience in gene therapy and serves as the PI on our FA patient natural history study and also is involved in many other FA studies. Our management team is made up of a number of biotech veterans with extensive experience in rare and genetic diseases. With our CEO and CEO recently coming from Sarepta, as well as Vertex Pharmaceuticals prior to that. And there are other executives coming from Genvax, Bluebird Bio, and BioBridges, among other places. We're all highly committed to using our technology to make a difference in patients' lives through discovering and developing meaningful new genetic therapies. Our lead program is FA. As you know, FA is a rare genetic disease, but it's the most common form of genetic or hereditary ataxia, with 9,000 individuals in the United States and also another 26,000 in the EU. FA is a recessive disease, which means that it leads to very low levels of a certain protein called frataxin. And frataxin is critical to making sure that the engines in the cell, or the energy producers in your cells called mitochondria, work well. When frataxin is low, protective iron-sulfur clusters don't form, and that leads to damage in the mitochondria and a lack of energy production that's harmful for cells that need energy the most. And those are ones like the ones in your heart and in your nervous system and brain and spinal cord, because they use a lot of energy. The generation of these cells is what leads to the symptoms of FA due to both damage in the heart and also in the nervous system. Unfortunately, to date, there is no approved therapies for FA. However, fortunately, through the work of patients like yourselves and physicians and also foundations such as FARA, there's increasing interest in developing new treatments for FA, and there are several companies now that are working on this, including ourselves. One therapy is even close to FDA submission, which is fantastic. However, we believe that multiple options will benefit patients the most, and that the best outcomes come from having multiple different approaches, such as gene therapy or even combining therapies. So we're excited about moving our gene therapy forward. We're taking a novel approach, which is to target both the cardiovascular or heart and neurologic symptoms using a dual route of administration. Our potential new therapy would be administered systemically in the vein or via an IV and into the cerebral spinal fluid via a lumbar puncture, which is also called an intrathecal administration. We think that splitting up the gene therapy administration will allow us to divide the dose appropriately to get into the heart, as well as to the brain and CNS and the rest of the nervous system for efficacy, while allowing us to have optimal safety. In addition, we plan to use immunomodulation as a pre-treatment. And this is based on the work of our founders, Dr. Byrne and Dr. Corti. This approach may help to reduce your immune response to the gene therapy when it's being administered, and we hope this will make it an even safer approach. We're making progress on our preclinical work, and we're developing a strong preclinical package to support an IND filing with the FDA so we can begin our clinical studies. We evaluated our research gene therapy construct in 3 animal species: rat, mouse, and non-human primate. And we were able to make 3 key observations. The first is that we were able to show that we could express the human protein in these animal studies. So that tells us that our gene therapy is doing what you'd hope it would do and produce normal frataxin. The other thing we were able to show is that not only was the protein being produced, which is great, but that this also led to improvements in heart function and survival in the animal models. In addition, the experiments that we did showed that we could improve the function of the mitochondria, again telling us that the gene therapy was producing frataxin that was acting in the right way to improve the situation. Thirdly, we saw no concerning safety signals in our preclinical work, including in our non-human primate studies, where we treated the animals with a similar regimen to that we expect to use for our first-in-human trial in patients. We're now performing some similar studies on an optimized gene therapy that we intend to take into the clinic. So what are the next steps? Our manufacturing team is working hard to prepare high-quality material that we would use in a clinical study. This and the preclinical and non-clinical work that I mentioned will be used to submit our IND, which should enable us to start our clinical study towards the end of next year. We're very excited about the potential to move forward into the clinic, and we look forward to providing updates over the coming year. Thank you very much for your attention, and also for allowing me to take a few moments to tell you about our company and our FA program. Thank you very much.
So if we're going to have all of our panelists speak, and we'll do questions at the end. Our next panelist is Jay Barth from Lexeo. And Jay, you want to come on up?
Thanks, Jen. It's really a privilege for me to be here among you and share with you some of the information that I can from Lexeo, the company of which I am the Chief Medical Officer. I'm Jay Barth, and I really hope to be able to convey to you what our plans are, where we're at right now in our program for gene therapy, and what the next steps are. Right. So I want to start by telling you a little bit about who we are. And Lexeo Therapeutics is a relatively new biotech company. It's based in New York City, and was founded by Dr. Ronald Crystal. For those who know the area of gene therapy, he's truly one of the pioneers in the field, having started in this area decades ago, and based in Cornell Medical Center in New York. And all of our programs, and I'll show you a little snapshot of other programs that we are undertaking as well, are all based on AAV gene transfer programs, which I will describe in a little more detail shortly. And we are focused in two areas, in the neurologic disease and cardiac disease. And that means both systemic dosing, such as intravenous dosing, and CNS dosing, dosing directly to the central nervous system. The pipeline that you see here, I want to show on top is the Friedreich's Ataxia cardiomyopathy program, and that is the one that I'll be speaking about. We are focused on gene therapy for treatment of the cardiac aspects of FA, and understanding that other companies are as well, or focused on the neurologic aspects, but our program is focused on the cardiac involvement, and I'll explain why. But we also have other programs in the cardiac area for gene therapy, as well as other neurologic disorders, including Alzheimer's disease and a rare neurodegenerative disease called Batten disease, as well as other programs that are earlier on. We are in the clinic right now in the Alzheimer's program and in our Batten disease program, and moving towards the clinical trial in our Friedreich's Ataxia cardiomyopathy program. Why are we focused on the cardiac aspects of FA? The reason is, as you, I think, well know, and as Dr. Lin explained before, the really high prevalence of cardiac involvement in FA, more than 90% of individuals having some form of cardiac involvement, it being a common cause of morbidity and mortality in approximately 60%. So we recognize the true unmet need that we hope to serve with this gene therapy. We think the heart is a very good target for treatment with gene therapy. Intravenous administration is an effective way to get the gene therapy to the heart. And as I'll show you shortly, we have the preclinical data, the animal data in mice, that show the effect of this gene therapy, which is the frataxin gene administered via AAV. We have non-human primate or monkey data showing good expression in the heart, and the fact that in a clinical trial, which we hope to get to, we will be able to assess the effects of the treatment through various assessments, cardiac assessments. I won't, because of Dr. Lin's excellent presentation on the aspects of cardiomyopathy and FA, I won't go into great detail, but just to make a few key points. The most common finding is the hypertrophic cardiomyopathy, mainly affecting the left ventricle. Systolic function, the pumping ability of the heart, is maintained for years, but over time, fibrosis occurs, dilated cardiomyopathy, where the walls of the heart get thinner and stretched out, may occur. Symptoms generally don't occur early in the disease, but occur later on. Arrhythmias are another complication of FA cardiomyopathy, and ultimately heart failure may occur. And because of all this progression of the disease that can occur, we really feel that a targeted gene therapy to treat the heart as soon as possible is our goal. So how will this gene therapy work? In the disease, as you heard from Jen, and this is an illustration of what's going on inside the cardiomyocyte, the cells of the heart. On top, in the condition of frataxin deficiency, the mitochondria, it disrupts iron-sulfur cluster biosynthesis, leading to interruption and disruption of many mitochondrial processes. As a result, the mitochondria, which are the energy producers of the cell, are not functioning normally. So the mitochondria don't produce enough energy, therefore the heart cells don't have enough energy. There's iron accumulation, other complications as well. So that results in the heart dysfunction. On the bottom, the name of the drug, LX2006, is the AAV, which is the viral coding. It's a non-infectious virus in which the frataxin gene is put inside the AAV virus, which is given intravenously. It makes its way to the cells, gets into the cells where frataxin is now produced. The frataxin is inside the mitochondria. It restores the iron-sulfur cluster biosynthesis. It restores the mitochondrial processes, and hopefully thereby restoring cardiac function. So this is the mechanism. And just a moment about the use of AAV-RH10. There are several different AAV viruses that are used in gene therapy now. This is the one that this program employs. It has good affinity, meaning it's directed well to the heart. It's also directed well to the CNS, although that's not the target with this therapy. And also, it has a low level of pre-existing immunity compared to some of the other AAVs. And that means there are viruses out in the world. People have antibodies to these viruses that could affect the efficiency of the gene therapy. So AAV-RH10, this type of AAV, tends to have lower antibodies circulating around, so people tend to not have antibodies to it. So I mentioned the mechanism of this gene therapy. We've actually shown in 2 mouse models that it is effective. On the left, in this slide, it is a very severe mouse model. In fact, it produces no frataxin whatsoever, more severe than any individual who has FA, where there's always some frataxin. And without going into the details in each of these panels, what is shown is that both for the size of the heart, the ventricular mass, the shortening fraction, which is the function of the heart, as well as survival, that treating these mice before they get the heart disease or even after the mice get the heart disease, it can be reversed. So this was the very encouraging mouse data that led to the program to take it forward. As I said, this is a very severe mouse model. This is a less severe mouse model that probably more closely reflects what an individual with FA experiences, which is cardiac symptoms that occur under stressful conditions, exercise, and so on. And again, with treatment, can restore normal cardiac function in these mice as well. So where are we at with this program? We have the mouse data. I did not show you the non-human primate, the monkey data, but it supports the fact that with intravenous administration, there's a good transduction. That means the gene therapy is getting well to the heart, so both in the mice and the monkeys. So right now we're finishing up those trials. We're heading towards an IND submission, as I mentioned, which is the step that's needed between the animal studies and going into human clinical trials. We're preparing for that phase 1/2 trial as we speak, with plans to start that trial next year in 2022 in adults with FA cardiomyopathy. It will be administered intravenously. The primary endpoint, as it is in all first-in-human studies, is safety, and that's first and foremost the most important consideration. But we'll also be looking at various assessments of cardiac signs, symptoms, biomarkers, and you've heard about some of those, the structure of the heart, such as the hypertrophy, looking at arrhythmias as well, and various measures of cardiac function so that we'll be able to assess the effect of the gene therapy. And thank you.
Thank you very much, Jay. Our next presenter is Doug Feltner from Execure Therapeutics. Doug, can I invite you up?
So I'm Doug Feltner. I'm the Chief Medical Officer at Execure, and I'll be telling you about the spherical nucleic acid technology and the treatment that we're developing for Friedreich's ataxia. Just a cautionary note that I'll be making speculative, forward-looking statements about the future, and this is required for me to include this. Can read it later, I guess. Okay, just a few comments on Execure first. We use this spherical nucleic acid platform technology. We're working on it for a number of genetic disorders. Friedreich's ataxia is our first CNS disorder. And I'll show you what that looks like and describe later in the talk specifically how we're going about approaching Friedreich's ataxia. There are therapies, and there are some on the market that use linear oligonucleotides. These are short stretches of synthetic DNA to alter gene expression, typically, and make more protein. What we've done is taken some linear oligonucleotides that are specific for Friedreich's ataxia and put them in the spherical format. What this does is it increases the cellular uptake, so we get a higher potency at the same dose. We've seen uptake in all neuronal subtypes, so there is broad CNS penetration. And we see that they last longer in the cells, so they persist longer in the central nervous system. We think we'll get prolonged efficacy because of that. We call the spherical nucleic acid we have in development for Friedreich's ataxia, Execure FXN. It's our intended to upregulate the frataxin protein, and that targets the molecular cause of Friedreich's ataxia. I'll go into some of these data in more detail in the talk, but we've seen up to threefold frataxin upregulation in a mouse model, indicating that there is a good potential to treat Friedreich's ataxia with this treatment. We've completed one safety study in rats so far, and it's well tolerated in that study. We're planning to do an IND by the end of this year and hope to dose our first patient in the first half of next year. Several people have mentioned INDs. That's an investigational new drug application. What that is we take all the information from the laboratory studies, cells in a dish, mice, rats, non-human primates, all the manufacturing information, we compile it all together in what's called the IND, and we send that to FDA, and we say, FDA, you should let us, go test this in humans, and then they get back to us, usually with some minor modifications, and hopefully we're on the road then. But the FDA, the regulators, do have to agree to going into humans. We have other projects going on. I'll just mention we have one in pain and one in a rare disease called CLN3 Batten disease that are right behind this project. And we have actually One project in the clinic called cavratolimod, it's a spherical nucleic acid that we're testing in a couple of rare skin cancers. And that one is in phase 2 right now, so we'll see how that goes. We recently announced a deal with a company called Ipsen in neuroscience to work on spherical nucleic acid treatments for Huntington's disease and Angelman syndrome, a couple of other rare neurologic diseases. So things are progressing well. Okay, here it is. On the upper left there is a picture of the spherical nucleic acid. It really looks like, a sea anemone or maybe a cush ball or something, but it has two components, and those are shown on the left. The synthetic linear oligonucleotides, so those are the strands coming out. That's the chemically modified artificial short pieces of DNA that can have therapeutic effects. And then in the center, that core is a benign lipid nanoparticle that kind of holds everything together. And it's this unique configuration that allows this to get into cells better, stay in cells longer, and we hope have more profound effects than some of the linear oligonucleotides alone. This is some early data we got from in another disease, looking at direct comparison between a spherical nucleic acid and linear. This is in spinal muscular atrophy. And I think, is this a... Yep. There we go. You can see this is survival of this mouse model. They're untreated. They pass away very quickly, about 20 days. If they're given nusinersen, which is Spinraza, it's a drug for a linear oligonucleotide that's used for spinal muscular atrophy. They live longer. If you give them the same amount of drug in this spherical nucleic acid format, they live much longer. So, and we think that's due to more of it gets into the cells, stays there longer. So where are we at in Friedreich's ataxia? The only thing, Jen and others have done such a good job of explaining, the basic biology. All I want to point out is the stuff on the upper left here. There's these extra GAA repeats in the frataxin gene. That's DNA in the nucleus. DNA serves as a template. It gets copied into RNA or mRNA. That's the message. That's carried out to the cytoplasm of the cell, and the RNA gets copied into proteins. Here we're concerned about the frataxin protein. And there's a deficiency, not enough frataxin protein, and that actually comes from not enough message in the cell because not enough messenger RNA is made. I'm going to skip that because that's been shown, but here's how the specific Execure FXN spherical nucleic acid looks and works. So in this SNA, we actually have two different linear oligonucleotides. We call them 1 and 2 here. They have different colors. And the first one, I have to give you a little background information. You haven't heard about this, but there's actually another RNA that comes from another gene in your cells in the nucleus that can block the making RNA from the frataxin gene. And so what our first one does, the first nucleotide, is it binds to that blocker and degrades it. So it removes the block. And that allows more of the frataxin mRNA, the message, to be made, which allows more frataxin protein to be made. So that's one of the two mechanisms. The other mechanism is mRNA stabilization. So here we have an oligonucleotide on the SNA that binds directly to the frataxin messenger RNA and stabilizes it. By stabilizing the frataxin messenger RNA, it increases the amount of messenger RNA that's there. and that increases the amount of frataxin protein. And so we have two methods in the SNA for increasing frataxin protein. And here's some data that shows increased frataxin protein and some consequences on mitochondrial function in cells. Now, these are cells in a dish in the laboratory. The cells are derived from patients with Friedreich's ataxia, so they have the genetic defect. And they're immortalized so they can grow a long time in the dish and culture. All right. So on the left, what you see is that untreated low levels of frataxin protein in cells derived from patients with Friedreich's ataxia. This axis here is the percent of the non-disease control. So again, it's 25-30% of normal. And you can see with increasing amounts of execure FXN, there are increasing amounts of protein in the cell. The second one shows you that that protein increases are in the mitochondria where the protein needs to be to work. And these are from two different donors, and we're just showing you one kind of intermediate dose here. But first, the lower levels are seen without treatment, and then we see an increase in frataxin protein in both of these two donor cell lines with our treatment. And then finally, what are the downstream consequences? So can we show something that says, hey, it's having an effect that ought to be important? And the answer is yes. What's measured here inside the mitochondrion is an enzyme that's important for generating ATP. That enzyme is called SDH, or succinate dehydrogenase. And ATP is that energy source in your cells. And so, indeed, we increase the activity of that enzyme that's important for generating ATP in both of these donor cell lines. Now, what about in mice? We do have this one experiment in mice. What we've done here is use a mouse model. And in this mouse model, scientists have genetically altered a regular mouse. They take out the mouse frataxin gene, and they put in a frataxin gene from humans from a patient with Friedreich's ataxia. And so this mouse carries the genetic defect for Friedreich's ataxia. And what we've done then is administer our execure FXN to these mice and then measured frataxin protein in different regions in the brain and spinal cord in the mice. This is a good mouse model for looking at frataxin protein levels. It's not so good for looking at symptoms or effects of the disease, because these mice don't have that many actual effects of the disease, but they do have the low protein levels. I misspoke there. So we haven't used the combined execure FXN yet in these mice. I have the results, though, of an early experiment that shows each individual component. So I showed you the component 1 and component 2. So those are the data that I will show you. And here they are. So what we've shown is that each of the two individual components do upregulate frataxin in mouse brain and spinal cord. In general, that regulation is 2 and up to 3-fold what the baseline levels are in the untreated animals. The cerebellum is a particularly important area, and you can see here's the level of frataxin protein expression in the untreated or vehicle control. And then with 2 different doses of the first component, you see these increases a little more than 2-fold, depending on dose. And then with the other component, a similar sort of story, dose response increase in frataxin protein in the cerebellum in a mouse. We do see increases, again, depending on dose, in the cortex, in the midbrain, in the brainstem, and then in various spinal cord regions. Cervical, that's sort of the neck of the spinal cord. Thoracic, that's your chest area, and lumbar, that's lower back there. So that shows you where we're at now with the animal experimentation in terms of looking at, can we increase frataxin protein in animals? We do think that we're well poised to move to the clinic. As I mentioned, we're hoping to have the IND at the end of this year and then dose patients, first patient, first half of next year. I want to mention working very much on our protocol right now and have worked a lot with a number of experts in Friedreich's ataxia, including David Lynch, who is here, and with Jen and Barbara from the Friedreich's Ataxia Research Alliance. I want to acknowledge their contribution and help. The primary purpose of these first studies, as you heard, is safety. And so the way they work is you start with a low dose, small number of patients, you test carefully. If things look okay, you give someone higher dose. Things look okay, somewhat higher dose. So we'll have a careful progression looking closely at safety. We do plan to include measures, both neuroimaging and biomarkers, to try to see that our drug is having effects that would be consistent with increasing frataxin in the brain and the central nervous system. And this is my last slide. On the left are some of the neuroimaging measures we'll be using. The first one, we're looking at levels of iron in an area of the cerebellum in the brain. The second one is some metabolites in that cervical region of the spinal cord and also in the cerebellum. And finally, a method, diffuser, DTI, that looks at the microstructure of the axons and the white matter that surrounds them. So we're hopeful that if we increase frataxin in different areas of the brain and spinal cord, that we could see some changes here as well. We'll look for frataxin protein in the cerebral spinal fluid, as well as some other biomarkers to try to see evidence that we're increasing frataxin levels. And one last thing, I just want to mention, the things that Friedreich's Ataxia Research Alliance does are really important for us and for developing treatments for Friedreich's ataxia. You can see that right here if you think about the TRACK-FA study. You know, they are pioneering a number of methods, particularly in neuroimaging and also in biomarkers with that study, that we're kind of piggying back along on to give us information for both, you know, designing our study and what we should measure, but also will eventually help us to interpret our data as well. So I'd like to just acknowledge that. That's it.
All right. Now next I would like to introduce Matt Kurth from Design Therapeutics. Matt's going to tell us about their new program that's coming along as well. And again, we're going to take questions at the end.
Thank you very much, Jen. Thank you for having us. Allow me to present some updates on our work. We're designing a novel class of genomic medicines that are actually small molecules. These are not genes, but rather these are molecular entities that you can synthesize in the laboratory. Let me see if I can get oriented. And by the way, I know it's tough to be the last speaker before break, but I'll do my best. To begin with, we are a public company, so we have forward-looking statements, and therefore we have to have our disclaimer. Notice the fine print, as my legal advisor always manages to squeeze more words in than necessary. All right. Our company is actually a relatively young company based in Carlsbad, north of San Diego. founded in 2017. We have about 75 employees, and we are developing a novel class of small molecules that are actually synthesized in the laboratory. And these are designed to treat serious diseases that are based around the trinucleotide repeat pathology. The lead program is on Friedreich's ataxia, and we've made some real progress, and I'm going to show you what we've done so far and where we're going. Our small molecules turn off or on individual genes and help target by being very specific to specific portions of the trinucleotide repeats. We call these gene-targeted chimeras or gene tags, and it's a unique way of looking and thinking about these molecules. Now, we've heard about the pathology of Friedreich's ataxia, and we know that the trinucleotide repeats in Friedreich's ataxia are at least greater than in the hundreds to thousands long. And this trinucleotide repeat expansion is like a block. It's like a gigantic stumbling block for the machinery that takes the DNA and turns it into RNA. And that's why in frataxic cells, patients with Friedreich's ataxia, the cells have a much reduced, about 25 to 30% level of frataxin protein in the mitochondria. In healthy individuals, there's no block, and you get normal expression of frataxin gene and protein. Thus, in normals, you have healthy mitochondria. In Friedreich's ataxia, the cells do not have enough energy because the mitochondria are impaired. Now, our drug normalizes the frataxin levels at extreme low levels and does it in a dose-dependent fashion. Let me get my pointer here and show you. So when you add the drug at When you add the drug at incredibly low concentrations, you can see that it quickly and rapidly, in a dose-dependent fashion, normalizes the level. What's even more exciting is the fact that this only works in patient cells that have the genetic abnormality. In normal individual cells, it doesn't affect the dosing at all. Now, how does this work? Well, keeping in line with the use of heavy words, our drug candidates are heterobifunctional molecules. And that means that they basically have two components: a DNA binding moiety that's highly selective for the GAAA repeat, and the ligand moiety, which then helps to overcome the block by helping the proteins that make up the polymerase activity to make RNA go across the repeats. So here's a schematic expression of how that works. Normally, the polymerase gets blocked, and these proteins don't properly march through that region. When our molecules are bound to the GAA triplicate repeats, they march across the thing, and they make the appropriate amount of frataxin. We have a QR code on our little flyer, and if you hit that QR code, there'll be a nice video illustrating this process in great detail. I won't go into the details, but the nice thing that's important is we don't have to have 100% of normal frataxin levels. We just need to get into the asymptomatic levels of those folks that have carrier status. Now, where are we in the progress of getting this drug into the clinic? We've already shown in cells that we have excellent potency and selectivity. And I have to make a real shout out to everyone who's donated samples. I want to thank Dr. Lynch and the whole FARA community who's helped collect samples that we actually use in our laboratory on a regular basis to test our drugs. And that is so important. Without that work, we wouldn't be here. We also use pluripotent stem cells, which we can differentiate into cardiomyocytes and neurons and show that our drugs work in that setting as well. The pharmacokinetic and distribution work is being done, and the drug has some incredibly nice properties in terms of duration of effect and staying in the blood and tissues. And this has been done in mice, rats, and non-human primates, little tiny monkeys. And I can never pronounce their names, so I'm not going to try. The non-GLP toxicology, which means it's done in a laboratory without meeting all the strict requirements of the FDA, has been done in mice and rats and looks really good. And now we're in the progress of putting together the package that's required to open an IND and have a chance to test the drug in the clinic. A couple of minor points I want to make is, actually these are really important points. We can restore the frataxin level mRNA by continuously exposing the cells to very low levels over a period of 14 days, and you see a nice increase in the RNA levels. And that's followed by a very nice increase in the level of frataxin protein. And this is done at 10 nanomolar, which is an incredible low concentration. That tells you how selective this molecule is towards improving the molecular defect of Friedreich's ataxia. Here we show that we're getting really almost to the normal level of frataxin protein, which, as you know, is above what we need to actually overcome the deficits of frataxin seen in Friedreich's ataxia patients. The other thing is, in our animal work in non-human primates, we're able to show that after after 3 doses, which is done at weekly intervals, so after 15 days, we have a continued level of the drug in the brainstem, cerebrum, and cerebellum above the threshold that we need of the 10 nanomolars to induce the frataxin protein. And indeed, sometimes it's even as high as 100 nanomolar. And this is true for the heart as well. So we have a drug that gets to the target tissue and hopefully will be able to induce the frataxin at intervals of treatment that might be at as little as weekly. So, safety. We've done once weekly dosing for 3 doses in Sprague-Dowley rats. We've also done it in non-human primates, and the data looks really good. We have had no significant systemic adverse events in these animals, and they've so well tolerated. And that gives us an opportunity to hopefully start clinical trials in the first half of next year. Thank you for your interest and any questions. And I just want to make a shout out to Wen, who's here. We'll be hosting a little booth out there. And we have the flyers in your program about our upcoming talk on October 25th that will go into more details in our clinical program and give you some more idea how the drug trial will look like. Thank you very much, and I'll leave it open for that.
Well, thank you so much to all of our panelists, and I hope that this really got you thinking and understanding a little bit more about how the work that we've done understanding the basic mechanisms can now be leveraged towards multiple different types of therapeutic approaches. And so we've got about 10 minutes before the break for some questions. Do we have any questions from the online audience? Not yet? Okay. What about questions from people in the room? If you have a question, just raise your hand. We've got mics. We'll bring them around. Randy?
Hello, Randy Ipe, FA Science Junkie. Wanted to thank you guys all for all you're doing for us and our families. It's critically important. One of the things I wanted to ask you, if you feel appropriate to comment on or can comment on, is if your plans involve opening clinical trials to a pediatric population, understanding this is the beginning stages of a pediatric disease, and if there's a chance to get ahead of the curve, it would be in the pediatric population. If there's any way you could comment on that, I think I'd appreciate that.
That's an excellent question, and we are, on a highly regulated business, and the FDA has very strict requirements for us to be able to go into the patient population. So we do have to start with adults first. The challenge is, of course, to have safety first, and if we can show excellent safety, given the condition that we're trying to address and the severity of the disease, we're hoping to negotiate with the FDA when we can get into the clinics with children as quickly as possible. I hope that answers your question in an indirect way.
It's a tough topic. I appreciate it.
Yeah, I'm happy to address that for Axicare right now, acknowledging that our protocol is not final. It's continuing to be in development. So things can always change. But we also plan to start with adults, and it's for the same reasons. It's for safety. Acknowledging that we would like to, once we have enough safety information in adults, we would like to move to including children and adolescents as soon as possible. I think we all believe that the earlier in the course of disease that we can treat Friedreich's ataxia, the better the outcomes will be.
I'll just add very quickly, I agree with what two other gentlemen said, and in our case, because the gene therapy is directed to the cardiomyopathy, apart from the safety reasons to look at adults first, it's really to treat existing cardiac signs or symptoms in adults, but the hope in the future is to go into younger individuals as well, because for the reasons stated, the earlier treatment, the better, and there it might take more of a prevention type of approach than a treatment approach.
Again, much appreciated. Thank you.
I think we have an online question.
So this question's coming from Mary Kearney in Ireland and Mary Caruso in Connecticut. And so opposite end of the spectrum from Randy, are there plans to include late-stage patients or patients who maybe have more severe cardiac disease in any of these trials?
I'll take that. We haven't defined yet exactly what the limits are for the cardiac disease. I think it will be a broad population of individuals. We do want to make sure in the first in-human trial, as it's called, safety being the main objective, that we are assessing the patients in whom there's the potential for benefit as well as the highest possibility of a safe outcome for them. So we haven't defined kind of the upper limit of what that would be. It may not include all patients with cardiomyopathy, but I think it'll be fairly broad.
I can add a little bit to that. In looking at first-in-human studies, we always look at safety first, but we also are interested in pharmacokinetics, which means how the drug is handled by the body, how it's absorbed, metabolized, and then excreted. And as long as the patients are stable in terms of medical conditions, there's no reason to exclude even more advanced individuals, and that's our thinking at this time.
Yeah, so we similar sort of comments. We will have some sort of exclusion on patients being too severely ill. Again, the protocol is still in development.
Allison?
Thank you. Thank you, Jen. As the wife of an FA patient, I just have to ask a question to all of you. I'm wondering if, as your companies work to enhance the delivery of frataxin and the efficacy of frataxin within the mitochondria, do you feel that in addition to being able to stop the progression of the symptoms of the disease, you may actually be able to reverse the evidence of symptoms already experienced?
Yeah, so maybe I'll comment on this. My last company I worked at was a company called Avexis that developed a gene therapy for spinal muscular atrophy. I think, this is a similar sort of story where you're trying to increase a protein. And I think what we saw was that, the earlier in disease you go, the larger the effects that you see. It can be difficult to tease out whether you're just stopping the disease from progressing or actually improving things. I think that with that question, it's really one that we'll have to see when we get in the clinic what we get. So I think we're hopeful.
I would just add from our standpoint, because again, it's the gene therapy directed to the cardiac disease, that the hope is to actually improve, not just arrest development. I won't comment on the neurologic aspects, which it may be different, but I think in terms of the heart and the mouse data that we've shown is that there is the potential for reversibility as long as there are enough viable cardiac cells present, which is generally the case, that you could actually see improvement. That's our hope.
Looks like we have a question in the back of the room.
How you doing? This is kind of piggybacking off her question. A lot of the slides showed, increases in call them key indicating factors. At this point, is there any data that shows how these will affect the symptoms of FA? as opposed to not just, increasing frataxin levels or other indicators?
I'll start with, again, directed to the heart, at least based on the mouse data, and that's the place where we can test it because the mouse models of the disease in this case have symptoms. They don't express symptoms, but they have low cardiac function. They're not able to exercise normally. And then with the gene therapy in the mice, they're then able to exercise normally and have normal cardiac function. So I think that's analogous, we hope, to humans of being able to show improvement in their exercise ability and other signs and symptoms of cardiac disease.
So, in the nervous system, the mouse model isn't great because it doesn't have much in the way of symptomatic defects. So we don't have data currently one way or another, really because of the absence of a good mouse model.
I have to second that. We'll have to see what the clinic shows. And I think the talk coming after the breaks might answer your questions from a more realistic perspective, because then we'll hear about clinical data, hopefully. So we're in the preclinical stage, and let's keep our fingers crossed.
Alicia, there's another online question.
Yeah, where are these initial studies planned to take place? Specifically, the question came from Amalia in Brazil, representing her 600 patients there.
Are we able to talk about where initial clinical trials are going to take place, or is that something you're still sorting out and working on?
We have not defined it yet, so I can't comment.
Okay.
But,
Well, it would definitely be in the U.S. Whether we go to other countries or not is still in consideration.
Given logistics, the U.S. is first, and we'll see what happens in terms of that. Otherwise, we may have more data by our October 25th talk.
I'll just say for, phase 1 clinical trials, you try and get through phase 1 generally as fast as you can, because you're primarily, addressing safety and dose. And what we've seen in the past with the sort of the development of other therapies for FA is that we'll start with small clinical trials in early stages, usually 1 or 2 locations, to answer those initial questions that then enable longer-term clinical trials that start to answer these questions about efficacy and long-term, how do the drugs impact treatment. And usually when we get to that second round of clinical trials where the studies are longer and larger is when we're also, able to expand in terms of number of sites and locations. And that's generally what we've tried to, work with our industry partners on to, trying to balance moving quickly and also, making sure that clinical trials are available to as many as possible. One more question, Jeremy.
Good morning, Jeremy Limpicki. Thank you again for coming and sharing your studies with us today. Assuming that Reata is able to gain their approval sometime next year, how do you see this affecting washout requirements for future studies? Or will patients be required to stop taking an approved treatment, or will this become the new baseline when looking at protocols and washout requirements for future studies?
So I'm certainly happy to address that, and it is something we've thought about some. Again, our protocol is still in development, but I think, if there's an approved therapy like the Reata treatment for Friedreich's ataxia, I can't imagine patients having to go off of it. That just wouldn't, I don't even know if that would be ethical. So once that's approved and it becomes available, if we have a protocol going on, we would have to modify our protocol. What we haven't, again, gotten to the specifics, but typically what you would see in something like this is that the patient would have had to have been on a stable dose for 30 or 60 days. And the expectation is that during the trial that, that they would continue on a stable dose. That's sort of typically how something like this would be handled.
All right, well, I know we're coming up to the break, And so please help me thank our panelists for participating today. Thank you for being here. We're going to take a break until 11:15, and I just also wanted to let people know that Susan Walther, FARA's Director of Patient Engagement, has worked really hard this year to bring new resources for the FA community on gene therapy. And so there's a webinar on the website. There's also videos. And so if this is all a new concept to you, I would encourage you to go and look at some of the new materials on gene therapy on the website to get more background and more understanding of what's going to be involved with those therapies. And so with that, we'll see you back in about 15 minutes.